we're going to go over like how we kind of assess and determine compost quality, soil health. One of the things that we're working on is bioassays, which are simple kind of potting mix experiments to test how well compost is germinating and growing plants. We're still kind of optimizing the bioassay. It does help to have a little bit more precision with it. So that's what we're kind of trying to work on right now. But you can see patterns already with the different types of compost. For example, vermicompost tend to see like germination happen very quickly. It's like the vermicompost treatments, generally things germinate first. So that's been interesting to see. Oftentimes the control potting mix is very slow, so the compost usually helps with the germination and growth of the plants. And we're trying to optimize it more because we want to get to statistical significance, so be able to do statistical analysis and show like significant differences between these treatments. And so that takes a little bit more precision to get that statistical power. And then we're going to talk a little bit about lab testing indicators. So what is compost quality? It depends a little bit on the end use. Mostly we want to create positive effects on soil health and plant quality. Sometimes with industrial compost or immature compost, it can suppress plant germination and growth or steal nutrients from it if it's not like well matured and well made. And so we want to make sure that we're creating beneficial impact from our compost production. There's a term called phytotoxicity, which is where the amendment is harmful to plant germination and growth and maybe potentially creates yellowing of the plant leaves and stems. It's also good for kind of generating consistency and troubleshooting issues, but also if you're at a garden or farm site, you generally can see pretty immediate effects just applying it there. But the bioassay, is a little bit more precise way to kind of determine what the different effects of different types of compost are. And I am always trying to think about how we can play with our feedstock and recipe and management process and understand how that impacts the end product and the compost quality and the effects on soil and plant growth. So being able to document from that start to finish is something that I'm really interested in kind of creating and refining. So we already do that a little bit, but it would be great to kind of bring it all together a little bit more tightly, more cohesively. So then we can experiment with our process and understand directly what kind of impacts that has on plant and soil health. So the bioassay setup, you use small pots. This one I have 1 gallon pots, but that takes up a lot of material. So I started using these little 2-inch pots. And you can change up the variables that you're testing for. So one is how much compost you're applying. The other is the type of seed that you're planting. So generally we choose sensitive seeds that are fast germinating, that are sensitive to the conditions around them. So things like radish, lettuce, or cress are good to use for bioassays. And we're still kind of testing out different seed types as well, like amaranth is one that we read about that we're interested in testing out. But any of those seeds work well in the bioassay setup. And then the different types of compost that you want to test, like fresh or immature, mature, sifted, unsifted, industrial compost, community scale, worm versus pile, whatever it is you want to test. Those are your treatment setups. So I talked about phytotoxicity. It's like basically any kind of harmful effect on plant germination and growth. So we did one bioassay at Cottonwood where we took, we used what was available on site, and that was these 72 cell trays. And I kind of, in order to get like repetitions or treatment reps, I kind of divided it into 5 quadrants for that whole cell. And so those were our kind of repetitions in that sense. So we tested a control potting mix, mature compost, fresh compost, and worm compost. And the worm compost was one of the first to germinate. Most of them did pretty well. The immature compost had less germination and growth in general, but things still germinated and grew okay in them. It was just like maybe a little bit lower percentage in that sense. So this is just a graph, some of the germination rates on these guys. This is our control mix, sifted mature compost. Vermicompost did well. The fresh compost you can see has a little bit lower kind of germination rate. And then the maturing end of the compost, which was unsifted and maybe not quite fully mature, but it's pretty similar to the rest of these, maybe a little bit lower. So I don't think we got like great statistical significance in the different treatments, but you can see kind of the general patterns. I set one up at Buena Vista to look at the Claremont compost and the Buena Vista compost. So our blue is the control potting mix, which is the lowest one. Our Quaker is the green. So this is doing the best out of all of them in terms of the germination rate. The Buena Vista compost is yellow here, and so it's a little bit variable. It's doing okay still, and then the immature is the orange from the Quaker meeting. So a little bit lower, but kind of similar to the Buena Vista compost. And that compost was like kind of mature, but not fully mature. So maybe it could still mature for a little bit longer, and then it could get up there. Also didn't have really great kind of statistical like differences here, but you know, again, you can kind of see the patterns where the mature compost is performing a bit better than the immature compost. So for each of these, to kind of make it kind of precise, we set up 5 replicate pots per treatment, and we try to plant about 4 seeds in each pot. Sometimes the seeds are really tiny, and like you get your, you know, variability where you accidentally drop like 2 seeds in one section or 3 seeds in one section. So those are some things that we're trying to kind of think about how to adjust for in terms of our being able to do like good statistical analysis on that. From what I've seen so far, vermicompost is usually really great. There's strong, quick germination. Usually I use about half as much vermicompost. So if I'm testing 20% compost application, I'll do vermicompost at like 10%. Immature compost, you know, still supports germination, but it's a little bit less consistent, lower percentage-wise, and we're still tweaking our details to get that statistical power that we're looking for. Romeo did a test of the Claremont compost versus a $10 organic soil. So this left section is the Claremont compost, and the right section is some $10 organic soil. So you can see practically 100% germination for the Claremont compost and like practically nothing for that store-bought soil. All right, so moving on to lab testing. This is going to be a little bit more expensive. You get a lot more information, but if you have a pretty consistent feedstock and management process, it's good to get a lab test, and that'll tell you, you know, what your quality might be like throughout your whole like composting. So you'll know, like, if I make compost this way, then it's probably going to be like this. If you're going to make major changes to your feedstock or management system, then you might want to get another test. So some of the indicators that I like to look at. So if you're looking for a lab to send your compost to for testing, these are maybe the indicators you'll want to look for. So they usually list like, okay, compost analysis, we're going to do all these indicators. So you could take this list and compare it to what they do and make sure like they have most of these indicators assessed. So the organic matter content, some of the water properties, which are listed as percent half saturation, that generally refers to the field capacity. General moisture content, C to N ratio, electrical conductivity, which is like the salt kind of content, total exchange capacity, which speaks to the nutrient storage, bulk density, which can tell you, it can be related to some of these things like EC, and organic matter content, and like just compost maturity in general. Nutrient content is good to take a look at it. So, you know, if generally compost is actually lower in nitrogen, so if you're wanting a little bit more nitrogen in your soil, maybe you want to add some amendments like feather meal or blood meal. And, you know, for industrial scale composting, they always want to test for pathogens like fecal coliform and salmonella. So we actually haven't done any pathogen testing on any of our community compost piles. So that might be something interesting to look at. But it's generally like a bigger concern for industrial facilities, especially for biosolids composting. So a lot of the regulations for industrial scale composting came out of biosolids composting, the temperature regulation and the pathogen assessment. So that's like some of the most risky kind of material to be working with, and so that's where the regulations have come from. Okay, so I'm just going to share some data from comparisons of different community composts from out here. So this is looking at the organic matter content. We have our LA compost, worm compost here. It's super high, like 85% organic matter, which is pretty cool. So worm compost generally tests higher. Courtney shared with me some results that she got, which were like 75% organic matter content. The LA Compost ASP system is a little bit lower, about 30 or so percent. The windrow from LA Compost is pretty high, 65%. This is our Quaker Meeting compost at about 45%. Amy's Farm is a little on the low end, 30%. This is the Huerta del Valle compost, and that's at like 40%. So generally, compost organic matter content is going to fall in the range of like 30 to 60 something percent for pile compost, and then worm compost generally tests much higher. So those are kind of the ranges you might be expecting when you're looking at your lab test results. This is the half saturation percentage. So this is the field capacity of water that these different composts can hold. So I was very proud of our Quaker Meeting compost. It was like one of the highest in the water holding capacity range. And everything else is also pretty good. The Amy's Farm compost was kind of low. And we tested this because they were dealing with the hydrophobicity in their soil. So they were wondering like what's going on with the compost that's contributing to hydrophobic conditions in the soil. And so that's why this has a very low kind of water holding capacity in the compost to begin with. So this is the salinity or the electrical conductivity. So if you have greater than like 4 EC in your soil, it's technically saline, so it's too much salt in it. So the Amy's Farm compost is like way too high. It's at like 20, which is in decisiemens per meter or so. And all of these are generally within range, under 4. This one looks a little bit high, but I believe they tested within, under threshold. But it's the Amy's Farm compost that's like way too high. So just like way too much salt content in there. And that was contributing to the hydrophobic conditions in the soil. We were thinking they were using too much manure in their feeds, in their recipe. And in the field, Klepto was already like starting to add more wood chips into their beds, maybe just like intuitively. And so the other thing is it could potentially be from the water. Like we didn't actually end up doing a water test, although Randy has said there is high nitrates in the water. And so potentially that could be contributing to the high salt content as well. And so he did adjust his recipe. So he's using more wood chips. And he kind of said that that really helped with the hydrophobic conditions in the soil. But I think there's still some element of hydrophobicity happening, so potentially it could be from the water. A look at the C to N ratio. So these are all fairly within range. Well, I guess the Huerta del Valle one is quite high. So it could be maybe not fully mature. Usually like really mature pile compost is gonna be within the range of like 10 to 15 or so. So like these guys are in pretty good range. The Amy's Farm is good for the C to N ratio. The Quaker and the LA compost composts are also within range. The ASP is a little bit high. I also think that this one was a little bit immature, and so that might be why it's a little bit high. It's over 19, it's maybe 20 something. Worm compost is a little bit different. It generally is higher in C to N, and so you'll see that it will be in like the low 20s, like 20 to 25 kind of range. So that's, if you see that, that's probably pretty normal. But for the finished pile compost, it generally should be within the like 10 to 15 kind of range. So another interesting pattern here is the bulk density. So you'll see that the Amy's Farm compost had a really high bulk density. Generally, your finished compost should be within the range of like 800 to like 1200 potentially. But this one was much higher, and so it was very dense and compact, and that was also contributing to like difficulty in facilitating water flow through the compost because it's like too dense. So you want some airflow and fluffiness in your compost so that it can facilitate water flow. The nitrogen content, you can see that the worm compost is super high in nitrogen. So we expect that the worm compost is much higher in nitrogen than the pile compost. The ASP might have been a little immature, so it has high nitrogen in it. Some of the other composts are a little bit lower. We generally expect that in pile compost. Like there's not actually like super high nitrogen content. It's more about the slow release of nitrogen over time through the biological soil food web. The Amy's Farm compost was a little bit higher in nitrogen. I think that kind of associates with the high salt content. So those were kind of the patterns that pointed us to the high salt potentially from the manure. These are summarizing some of the issues we saw in the Amy's Farm compost. Low in moisture, organic matter, excessively high in EC, high bulk density. So salinity is an issue. It can limit beneficial organism growth. You can recognize saline soils by like spotty growth and white salt crusts. And as the soil and compost dries, the salts concentrate and accumulate over time. So it makes less water available to plants as well as nutrients. And plants have different abilities to tolerate salts as they grow. And leaching is really the easiest way to deal with salts in your soil. So that's what they were doing here. They would just have to excessively kind of water the beds in order to leach the salts from the soil and allow the water to infiltrate into the soil. So it uses a lot of water to deal with that. So probably too much manure. Definitely adjusting your recipe really helped, but potentially there might still be salt nitrate contribution from the water. So it's another thing to potentially test in the future. Some of the local labs we go to are Waypoint in Anaheim, Wallace Labs in El Segundo, Earth Fort Biological Testing. So just make sure whatever lab you go to that they test those different indicators. So for biological activity, we could do a general microscopy overview, and that's what we're kind of doing at LA Compost. With the general microscopy, you can at least see, relative abundances of bacteria, fungi, protozoa, nematodes. Are those different organisms represented? Can you see them? And what's their relative abundance? So you can see for like a well-made, mature compost is a well-rounded representation of all these different groups. Like a really dry, like poorly managed compost is only like has bacteria in it. You can't really see anything else. So those are some of the patterns that you can observe just from a general microscopy kind of overview. Worm compost generally has more fungi in it, so I generally do see like more fungal threads in worm compost. I really want to try to help to develop kind of overall documentation systems. We are going to do phase 1 software tool, which is mostly going to be inputting your diversion data and temperature data. But we want to develop it so that we can input our maturation time and be able to integrate information about compost quality, such as your lab testing bioassay or microscopy assessments. Pair that up with detailed information about your feedstock, turning and management temperature, and really be able to see like what feedstock management processes correlate to what kind of quality indicators. So that's something that I would love to see kind of develop from the software tool that's going to be launched in this program. All right, so I'm going to move into soil testing. So some of your sites we're going to do pre-test, post-test. It might be like an empty site, so we do pre-test. And then as you do soil building activities after about a year, we'll do another post-test and see what's going on there. And that helps to showcase, how our soil building activities, give evidence that our soil building activities are actually improving our soil biology and soil health indicators overall. So we want to test for heavy metals, different health and other characteristics depending on how we're using the soil. So we want to test when we're coming into a new site, and then after we're building the soil. If we're trying to maintain soil fertility for vegetable farming, we want to know a more detailed mineral breakdown of what's happening. We probably want to do a soil test to see what kind of amendments we might need. And if we're coming across any issues, then we might want to do some soil testing. So a lot of similar indicators for soil testing as the compost, the organic matter content, available water capacity, pH, EC, cation exchange. The Cornell University does aggregate stability, so you can actually get a sense of your soil structure, which is pretty cool. Not everybody does that. Cornell University also does soil protein assessment, so that's your organic source pool of nitrogen. So instead of the pure plant available nitrogen, it's looking at your protein content. So they call that the potentially mineralizable nitrogen, so the nitrogen potential that can be provided to plants. So that's kind of a cool ecological kind of indicator. They also look at soil respiration, so the CO2 content that's coming out of the soil, which is associated with the microbial activity. If we are able to do biological testing, then we can assess the fungi to bacteria ratio, if that's something that we're trying to cultivate. Is if we're trying to encourage a greater fungi to bacteria ratio, then that's something we might want to look at. Beyond that, the whole soil food web, there's different kind of indices for nematode community that we can do as well to assess the maturity and stability of the ecosystem. And then the mineral content, actual mineral content, is also important for vegetable farming, but it's very much related to the biology and ecology of the system. So it's not the only thing that's important, but it is good to look at it as well. I guess I'll mention high throughput genetic sequencing. This is more of a research-oriented technique, but basically you can take the DNA from soil or compost and sequence it to understand the microbial community composition, and you can do this for all organisms. It's not very commercially available, but the idea is that it's more accurate representation of the microbial community composition than laboratory culture or even microscopy, because it's really looking at the genetic information there. So there's different ideas about that, but it does give you a really broad kind of breakdown of the different organisms that are there. So that's a technique that I used in grad school and previous work in research, but it's not very commercially available. Although we are interested in trying to set up our own little sequencing lab. So hopefully we can do that. We would need some help if a lab was shutting down and wanted to give away all their laboratory equipment. We kind of need that basic laboratory infrastructure. So that's something we would be looking for in order to be able to set that up. But Stephen at Lopez was down to let us use a shipping container on his site to set up a little sequencing lab. So that would be really cool. You could send us all your samples and we could do genetic sequencing to give you an output of your microbial community composition. So yeah. Another test that can be done is looking at the mycorrhizal colonization. It's more of a microscopy technique where you stain the mycorrhizae to see them. And then another research technique is to kind of look at the enzyme activity in the soil. So that speaks to what kind of nutrient metabolic processes are happening. So it's kind of an integrative indicator, and it could speak to what organisms are active, what nutrients are being digested. So that's why enzymes are interesting to look at. Soil testing labs. So I do really like the Cornell Soil Health Analysis Lab that is in the East Coast, but they do that kind of more ecologically based overview of soil health with like the soil protein, soil respiration, and the aggregate stability and available water capacity. So I find that to be really cool that they do those indicators. For heavy metals, it's good to send to Wallace Labs out here. If you guys have questions about labs in your area, you can ask me about that. Waypoint is local to here in Anaheim as well. If you're really looking for like kind of agricultural mineralogy, Logan Labs in Ohio is kind of good for that, and there's like some techniques developed by based on the work of a soil scientist, William Albrecht, where you can kind of calculate based on your mineral content what types of amendments you need. So Logan Labs is good for that. All right, so just share some data from pre-test, post-test at Buena Vista. I think I just got the Earth Fort results back and didn't have a chance to put it into a graph, but this is the Wallace Labs results. So our blue bars are year 0 at vacant lot status. The green fluorescence is like last week that I sent those samples in. So it's been like a year and a half of soil building activities there. So you can see it's like the mineral content increasing by like many times, 2 to like 5 times increase in the mineral content of the plant available nutrients. And it is a lot of, it's from all the organic matter that we loaded there, but the organisms are kind of mixing up the native soils with the organic matter together. This is the water properties. The blue is our vacant lot, and then the green is our right now situation. So the field capacity from the water has increased like 4 times. Moisture content has increased like 4 or 5 times. The organic matter has gone way up. And so this is our first year, so organic matter is going to be more high because we just loaded fresh material there. But as it develops and matures, like next year, like generally for no-till vegetable systems, I've seen it to kind of like reach a threshold of maybe 11 or 12% organic matter. So that's where I would expect to see it kind of balance out over the next couple years. This is a different project at the LA Arboretum where we looked at different types of mulch and their effects on the soil. And this one we sent to the Cornell University, and they were able to do the aggregate stability tests. So we have our unamended soil on the left, we have a lasagna mulch in the orange, and then we have a pure bark mulch in the purple, and then a hugelkultur on the right. And it's kind of cool that the pure bark mulch tested the highest for aggregate stability and was also the highest for water holding capacity. And in my grad school experience, it was the hardwood bark compost that was most suppressive of disease, aside from the worm compost. So it's like another kind of lends evidence to the fact that different types of organic matter have different characteristics and different functions and effects on the soil. So it's like the bark is like the living skin of the tree. So like the inside of the tree is like more structural. It doesn't have as much biological activity. And it's more like structural for passing like fluids through. And then the bark is where all of your kind of microbial activity is to protect the tree from pathogens on the outside. So potentially it's like the higher biological activity on the bark and potentially fungi that's contributing to better soil structure, better water properties, and disease suppression.